Today I'm going to be talking about transdiagnostic imaging of a particular protein called the alpha-7 nicotinic acetylcholine receptor. I have no disclosures. I'm a clinician, so I just wanted to start off by telling you and underscoring the fact that medicine as a whole is undergoing a paradigm shift, where the science and art of medicine is rapidly changing, I think, for the better. On the side of art, I think of the movement toward value-based healthcare, how we measure value, how we improve upon it. And then on the side of science, there's an emphasis on precision medicine. Precision medicine is using data to subtype individuals with a given condition to get the right treatments to the right patients at the right time. And it's not a new concept. So precision health was talked about by Sir William Osler. He was one of the early faculty members at Johns Hopkins University. He said, variability is the law of life. And as no two faces are alike, no two bodies are alike, and no two individuals react alike and behave alike under the abnormal conditions which we call disease. So, of course, right, two individuals with bipolar disorder could come to me and present very differently in their symptoms. And I could give them lithium, and one patient might react very differently than the other. And I think it's very important that modern medicine keeps that in mind. So that brings me to today's talk. I'm talking about positron emission tomography, or PET-based precision brain imaging. And what I mean by that is using imaging agents specifically targeted to key proteins in the brain to subtype patients with a given condition by their molecular brain signature. And of course, the goal of this is to get the right treatments to the right patients. So it breaks down, we have an imaging agent, we call that a radio tracer on the left, and then in the middle we have a detection device, the PET scanner, and then we model the data to get a readout on where those proteins are in the brain and whether they're changed in disease. Our group is committed to developing new radio tracers to light up different proteins that we think are relevant to neuropsychiatric diseases. But today I'm going to focus on one protein in the interest of time. So today, again, I'm going to talk about the alpha-7 nicotinic acetylcholine receptor. It's an ion channel. It's expressed on neurons as well as some other cell types in the living brain. And in evaluating whether this receptor is changed in psychosis, we're really just revisiting a decades-old hypothesis. So Bob Friedman in the mid-90s used post-mortem brain tissue and showed that this receptor is low in the brains of cases with schizophrenia. Marty Pomper, with whom I work, showed the same thing in separate cases of schizophrenia. And interestingly, Dr. Pomper did not see that pattern in bipolar disorder. And you'll see that we're keeping these early results in mind as we image the living brain. The other key background piece that I want to emphasize is that there are medications, both existing and emerging medications, that target this receptor to increase its activity. So if we can identify individuals with low availability, perhaps low activity of this receptor, perhaps we could get these medications to those individuals. Many of these medications are being put forth for helping with cognitive deficits in neuropsychiatric diseases, including schizophrenia. So we think our data are going to be important to guide the use of these medications. Okay, so our imaging data. We have this new radio tracer to light up this receptor. The way that we start out is we image baboon brains and show that the imaging agent is both safe and effective at imaging our target. Then we move into healthy human brain imaging, and then we've recently studied across the adult aging population to show that this is something that even elderly, frail individuals can undergo safely. And in that study of aging, we learned that age matters. So we saw an interesting pattern that the receptor's availability seems to increase with age. We don't completely understand that. We're pursuing it further. But going forward, we're always adjusting our results for this potential effect of age. And then this is the data that I want to spend some time on. So this is using F18 ASEM PET to image this receptor in recent onset psychosis. I'm defining recent onset as within 5 years of onset of psychotic symptoms. And let's focus on the left figure first. So this is data from hippocampus, that structure that Dr. Kai was talking about that's involved in memory. I have the data divided into 3 groups: healthy controls who do not have mental illness. This middle group is patients with bipolar I disorder. So remember, that's when psychosis occurs during either mania or the depression. I lump those, I term them affective psychosis, or AP. That's why you see the AP here. And then the third group is patients with schizophrenia and schizoaffective disorder. I lump them as a non-affective psychosis, or NP, because the psychosis can occur independent of mood state. So as you can see, the 2 patient groups, both affective psychosis and non-affective psychosis, on average have lower availability of the nicotinic receptor compared to the controls. But that low availability of this alpha-7 nicotinic receptor is really most robust in those with non-affective psychosis. And then if you look to the right-hand figure over here, I'm showing whole brain images, and you can see that this finding is not just limited to hippocampus, it's really across the brain. Another way to look at those data is to lump all those patients together to look transdiagnostically, and to see whether that low availability of the alpha-7 nicotinic receptor tracks with certain signs or symptoms of psychosis. And that's what we've found to date. So among patients, lower availability of the alpha-7 nicotinic receptor in hippocampus is associated with lower cognitive performance in certain domains, so processing speed and verbal memory. And we're going to pursue that further, which takes me to next steps, which are possible because of your generosity. We've designed a larger study to expand this imaging into many more patients with recent onset psychosis. In that larger study, we can look at the association between certain signs and symptoms of the disease, as well as start to parse out effects of medication and sex on our imaging. We can also look a little bit into why, right? So we're going to get DNA from each patient to see whether there's a genetic underpinning behind our findings. And then we're going to collaborate in parallel with those who are studying the underlying biology of alpha-7 nicotinic receptor signaling, because of course it's important to understand the biology. All of these data are hopefully going to inform those clinical trials with those medications that I talked about up front. And of course, the ultimate next step, right, is to reduce this knowledge to improve clinical practice. And I think it's really timely work, it's important work. I can't thank you enough for your support of it, because this is the time science and art in medicine are marching in this direction of high-value precision care. And it's my opinion that beyond the clinical interview, diagnostics, including PET, are going to become more important as the clinical value of subtyping conditions is recognized. So thank you very much for your time. I should thank my collaborators as well, but I'm happy to answer questions.